right, lipids. In this section, you should be able to compare the structures of fats, phospholipids, and steroids and state the function of each class of lipids. So this macromolecule is lipids. Lipids are going to vary in structure quite a bit. They are going to be very large nonpolar molecules that are insoluble in water. This is going to be due to their large hydrocarbon chains. These are going to be hydrogens that are covalently bonded to carbon, are nonpolar, and tend to not form hydrogen bonds with water molecules. They're going to be diverse in their function and forms. They're going to be found in the forms of triglycerides, fats and oils, phospholipids, and steroids. So triglycerides. As I may have mentioned, fats and oils, lipids do not form polymers. So we're not going to have very long chains of subunits all sort of strung together, repeating subunits strung together, but we are going to have we're going to have a lot of similarities in form and function. So in this case, instead of a whole lot of chains all strung together, what we're actually going to have is a backbone molecule and then chains kind of hanging off of that, rather than a bunch of boxcars strung one right after another. So this is going to be our triglycerides. They're going to be made out of 1 glycerol and 3 fatty acid tails. Each fatty acid is going to consist of a very long hydrocarbon chain with an even number of carbons and a hydroxyl group at one end. They're typically going to be used for energy storage, insulation, and cushioning, and come in 2 forms, fats and oils. The fats are going to be usually from animal origin. They tend to be solid at room temperature, whereas oils usually come from plant origin. They tend to be liquid at room temperature. So here is how we can actually synthesize our triglycerides. As with all of our other monomers and polymers, our large molecule, our triglycerides, are going to be made up of a glycerol molecule and 3 fatty acids, and they're going to be strung together using a dehydration reaction. We're going to remove an H from each head on this glycerol molecule, and we're going to remove an OH from each one of these fatty acid tails. So when we dehydrate, we are going to attach this 1 glycerol to 3 fatty acid tails to produce 1 fat molecule and 3 water molecules. If we then want to break down this fat molecule, we're going to have to do this with a hydrolysis reaction. It's going to require 3 water molecules in order to break each one of those fatty acid tails off of that glycerol. So, as I stated, triglycerides are hydrophobic. They tend to clump together in body fluids. Emulsifiers are molecules that surround triglycerides and disperse or emulsify them. This is going to become important in our later chapter when we talk about digestion. This is going to allow enzymes to fully break down the triglycerides by emulsifying them in our digestive juices. So another very important lipid that we want to talk about or mention is waxes. Waxes are going to contain 1 fatty acid attached to alcohol. Their primary goal is going to be to prevent the loss of moisture from the body, specifically body surfaces like the cerumen or your earwax. Saturated and unsaturated fatty acids. The fatty acid chains in triglycerides could either be saturated or unsaturated. Saturated fatty acids have no double bond between their carbon atoms, so they contain as many hydrogens as they could possibly hold. So they are saturated with hydrogen atoms. Saturation refers to every carbon being completely saturated with those hydrogen atoms, and they tend to be solid at room temperature. Diets that are really high in animal fats have been associated with circulatory disorders because of the saturated fats that can accumulate in the blood vessels and sort of clog them up. Unsaturated fatty acids contain double bonds in the carbon chain. This is ultimately going to reduce the number of hydrogen atoms present in the chain. Additionally, the double bonds may have chemical bonds on the same side. They tend to be liquid at room temperature. They're going to melt at a much lower temperature. And the reason for this is because that double bond actually creates a kink or a bend in that carbon chain. It prevents a very tight, close packing of those molecules. Nearly all animals use fats as their long-term energy storage, and they tend to store more energy due to those carbon-hydrogen bonds. So here's just sort of an example of what that looks like. So here is our single bonds of our saturated fatty acids and our unsaturated fatty acids. Our unsaturated do not contain as many hydrogens as a saturated bond. This double bond can actually cause a slight kink in the tail. It'll cause a little bend in that tail, and it prevents these tails from packing too closely together. So this is how they maintain sort of a liquid at room temperature. Hydrogenated oils or hydrogenated unsaturated trans fats. This is where we can actually add hydrogens and reduce the number of double bonds in unsaturated fatty acids. So things like shortening and margarine. We can actually reduce those double bonds, or we can sort of unkink that bond by placing these hydrogen ions or atoms on either side of the bond. The hydrogenation increases shelf life. It makes solids fat, or makes fat solid at room temperature. So that's why we call it trans fat. But it may also be associated with a greater risk for heart disease. So the next important lipid that we're going to study is the phospholipid. It's very similar to the triglyceride, where we have a glycerol molecule with those fatty acids hanging off the edge. So this phospholipid consists of one glycerol molecule linked to two fatty acids and a modified phosphate group. So this portion of the molecule is known as the phosphate head of the molecule. This group, this region up here, is polar. So if this region is polar, does that mean that it is soluble in water or insoluble in water? It is actually readily soluble in water. It's going to be hydrophilic. We have a hydrophilic polar head. It's going to have a huge affinity for water. Unfortunately, or not unfortunately, but conversely, the nonpolar tails, these fatty acid tails, are nonpolar, which means they're hydrophobic. They do not like water. Now, because of that, we're going to see these hydrophilic polar heads are going to orient themselves to the inside of the cell and the outside of the cell. The inside of your cell is mostly water. The outside of your cell is something we call interstitial fluid, which is, again, mostly water. The hydrophilic polar heads like the water inside the cell and the water outside of the cell. This causes them to form a bilipid membrane where these phospholipids will actually line up. So we have a water soluble side on inside the cell and a water soluble side outside the cell. And what we have in the middle is sort of this non-polar valley where all these non-polar hydrophobic tails like to hang out. Another thing I would like to mention is that one of the tails has a double bond. This causes it to kink ever so slightly. This prevents the super close packing of these phospholipids, and it helps maintain the fluidity and the rigidity of the structure at changing temperatures. So phospholipids, like I said, that structure is very similar to a triglyceride, but one fatty acid is replaced by a polar phosphate group. They have a polar phosphate head and a nonpolar hydrophobic tail, and it's going to be the primary component of the plasma membrane responsible for forming that lipid bilayer. The last type of lipid that we want to discuss is going to be steroids. They're a huge part and very important part of the lipid family, but they have very different structures than triglycerides or phospholipids. They are going to be composed of 4 fused rings, carbon rings, and their various functional groups attached to the carbon skeleton are going to be responsible for their different functions. One of their primary functions of like cholesterol is that it's going to be a very important component of the animal cell membrane. There are a lot of different steroids. For example, testosterone and estrogen. Testosterone and estrogen are sex hormones. They differ only in the functional groups that are attached to the same carbon skeleton. If you look at this, we have testosterone, cholesterol, and estrogen, all of which look very, very similar. The only difference being the number of double bonds found on these first 2 rings and some of these functional groups that are attached to the outside. Cholesterol is going to be the precursor molecule for several other steroids. So cholesterol is actually a very important part of our diet. It's a very important part of our body. We require it as a precursor for our other steroids. It also plays a very important part in the cell membrane. All right, and that's it for our discussion on lipids. You should be able to state the functions of triglycerides in the human body. You should also list the uses of phospholipids and steroids and summarize why not all lipids should be avoided in the diet. You should know the significance and the importance of these lipids and why we have them in the first place.